So it's 2 o'clock on the East Coast of the United States. It's C-Star time. Welcome, everybody. Welcome to our audience. Welcome to our speaker for today. Before we head into the introduction of today's speaker, I want to alert you to the last C-Star lecture of the series, which is of this season, I should say, which is in 2 weeks' time by Yasmin Farooqi Shah from the University of Maryland, and she'll be talking about grammatical encoding in aphasia. What do we know and how do we move forward? That will be the final talk of this particular season, and then we will start up again in late August with our next series. So we're already planning people's talks and speakers. So if you have ideas for that, or if you want to give a C-Star talk, feel free to contact us. My email is on the screen right now. During today's talk, if you can ask your questions via the chat box, that is much appreciated. You can start doing that during the talk, and I will read those questions out after the talk. Today's speaker, we're really excited to welcome Dr. György Buzsáki, and he will be introduced by the PI of our arching C-Star project, Dr. Julius Fredrickson from the University of South Carolina. Julius, it's all yours.
All right, thank you, Dirk. I'm delighted to introduce Dr. György Buzsáki. He's the Biggs Professor of Neuroscience at NYU School of Medicine. György got his MD from the University of Pécs in Hungary and his PhD in neuroscience from the Academy of Sciences in Budapest. György's primary research interests focus on the mechanisms of memory, sleep, and associated disorders, as well as neural syntax, or how segmentation of neural information is organized by brain rhythms. I first became aware of György's work when I read his book, Rhythms in the Brain, but my motivation for asking him to give a C-Star lecture was because of his most recent book, The Brain from Inside Out. And I would say that I'm an avid reader, but it's very rare that I read a book more than once. But I've already read The Brain from Inside Out twice, and from time to time have looked at it again just to skim some chapters and remind me of the main concepts. The reason why I really like this book is because it proposes what I consider to be a very sensible approach to thinking about how functions are organized in the brain, which I believe has implications for understanding the neural mechanisms that support speech and language. Before I give the word to György, I want to note that last year he had almost 10,000 citations, which I've never seen in the field of neuroscience before. Well done. In any case, György, we're honored to have you with us, and I'll turn the presentation over to you. Welcome.
Thank you, everybody, and thank you, Julius. Well, we have a couple of things in common with you, namely that we believe in the primacy of action. And this is basically what my talk is about. But I'd like to introduce this entire topic with three sentences or three questions. The first one is what is an interesting one, and I think most of us can think about it and should think about it, is that we are working with a vocabulary, and we would like to know where that vocabulary has come from. And I will give you my version. Then connected to this, I'd like to address another issue, is that when throughout the evolution, we go from simple to more complex, and we would like to know where the cognitive mechanisms that we all have as humans come from. Obviously, that's a speculative part of my talk, but I'm willing to stick out my neck and see what happens. And the third one that is somehow connected to that, perhaps to the first one, is that nobody in this audience believes that the brain is a blank slate or a tabula rasa. But the reality is that we all organize our experiments and interpret our data according to this model. And if this is not a good model, what else is available? So let's start with the first question. And of course, the common denominator of all of this is what we call action. And you will see what I call and how I define action. So when we ask ourselves is that, what are we doing in neuroscience? And the answer is that we pick a term, let's say motivation or emotion, language is one of them, or many of these things. And we are saying, okay, this is an interesting topic for myself, and I'm going to devote my entire life researching the brain mechanisms of it. But then when you ask us, what are the relevant connections between those things that we already know and inherited from our predecessors, and how are we trying to understand the brain? And the truth is that we didn't start out trying to understand the brain. We started out trying to understand the mind, and especially at the beginning, the soul. So this gave rise to a philosophy that we still use and have and work accordingly, that there is an input, and that's the world. And the goal of the brain is to absorb the truth of the world, evaluate what happens out there, and that evaluation has to be coming to a deliberation, and then we decide whether we act upon the perceived item or not. And this is what I can call input-output. You can call it action-perception cycle, and there are many ways doing that. Or in a general framework, you can say outside-in, which means that indeed the goal of the brain is to perceive the world the way it is. Well, as I say, there are complications with it. The important complications that I faced early on in my career, and all of you had the same problem, is that how is this transformation made? And we have not much choice but to invent something in the middle or in between input and output and say, oh, if you're an engineer, you can say it's a black box. If you're a psychologist, you can call it an intervening variable, so information processing. You know, these are two words that I don't understand. I don't understand what information is, and I don't know what processing means. Or representation, so you can say model of the world. Or consciousness, we can go up to homunculus and all the way to free will. And of course, if you would like to submit a grant today to say, I'm going to study free will to NIH, chances are pretty low that you will get funded. But if you submit a grant with decision-making, no problem, you will get the grant. And the truth is that they are exactly the same. There's not much difference. And that's, you know, we can argue back and forth why it is not so different. So the alternative thing to think about the brain is that the brain is not interested in the world. The brain's primary preoccupation is to maintain its own dynamic. That's the primary thing. The secondary thing is to generate an output, because without output, without action, without movement, no creature can survive. And once you have an output, then you would like to adjust that output or optimize that output. And this is why you need sensors. So the sensors come in evolution as the third level. So I call this framework the action perception cycle, if you want, or the inside out prediction. Now, these two different views, of course, come with different connotations and different things, because if you are thinking about that you have to synthesize the input, then you have to struggle with things such as in the middle, as I said, consciousness, homunculus, and so on. But also is that, how do you learn something that is specific? And you can say, oh, there are many, many specific things, and you generalize, this is the binding problem. And then other consequence of this outside-in view is that it's a representational view. You think there are things that are equivalent things in the brain coded in a spike format or other neuronal formats that represent something out there. The other aspect of it is that, of course, it's not necessarily the case. In fact, we can go the other way around. For a newborn, nothing is specific, everything is general. For a newborn, there is nothing unknown. I cannot show you or anybody something that we say doesn't exist, because the brain always generalizes, and then it goes from general to specific only when there is advantage to the organism of knowing something more. So this is what I call the first thing that most of the brain circuits do, is a good enough solution. And then if you'd like to get specific and you would like to get more information about something, this is when you go in and use very large part of your brain real estate. Okay, so this is a very, very general introduction, but I can be a little bit more specific by saying that the idea about this outside in actually happened, that started out perhaps in, if you look at just the Western hemisphere with the Greeks, and then it happened in the Christian philosophies, where of course it was a goal to understand the truth, because the only thing that we are interested in is how to choose between good and bad, and we have to choose good, which is God, and then evaluate and act accordingly. And of course, this ability to choose between bad and good was given only to, not humans, only to man. But then over the centuries, this Christian philosophy was adopted by the British empiricists, and fast forward to 1819, when this extraordinary book by William James, The Principles of Psychology, was published. And this reflects basically the philosophies that many people agreed upon, especially the empiricist philosophers. And the interesting part of course, is this books, that there are these chapters, and most of the chapters, almost all of the chapters, are dealing with the inputs and a little bit in the middle, but there is very little about the outputs. But if you look at this again, then you can say, I can pick a word from this series, I call it James List, and then you can spend the rest of your time trying to figure out, what is the mechanism of association, what is the mechanism of discrimination, comparison, imagination, and so on. But the paradox to me is that these words, as I said, was concocted and made by people who were trying to figure out the mind. In other words, these things are not grounded, but this list of things were given to us as neuroscientists with the goal that what we have to do now is what we have to do as a field is to figure out the brain mechanisms of these ideas that were imagined by people who had no clue or didn't even care about the brain. And I will show you that there are major flaws with this. So over the years, of course, this number of chapters get multiplied, and the words and the terms that we use in neuroscience got multiplied, and many things were put into various places in the brain. In other words, we were looking for homes for the terms that we made up. But we were diligent enough, and we made many, many terms. And as you all know, when it comes to about memory, you say is the hippocampus. When it's about fear, we say amygdala, and so on and so on and so on. And if we don't know where to put things in the brain, then we put it in the prefrontal cortex. And this is why there's a whole long list of the prefrontal cortex terms. I just put a few of them here, but there are many more. And it's very unlikely that this piece of cortex does so many things. And what are the relationship among them, such as consciousness, mimicry, goals, working memory, and so on. And of course, the main problem is that there are people who are working on these words, they are working in different laboratories, and there is very little agreement across laboratories. So why is this a problem? The problem is that my claim is that this way we will never understand how the brain learns about the world. I know it's a very strong statement, but let me try to explain. So this is the typical paradigm that many laboratories, including mine, have been doing for a long period of time, which is, this is the famous Hubel and Wiesel cartoon, that is, we give inputs to the brain, and we are looking for responses in the brain, such as unit firing, bolt signal with the fMRI, hormonal changes or chemical changes, and so on. And then we find something interesting because we find a correlation. Then we claim and write in a paper that we found the code. We found the code of face perception. We found the code for sound identification and so on. Now, the problem with this is that this correlation makes sense only to one person, who is the experimenter, because it is the experimenter who has access to the outside world as well as what happens in the brain. In contrast, neurons in the visual area have absolutely no clue what they are seeing. Neurons don't see the world. All they see or all they feel is the actual potentials coming in from other neurons. In other words, if you make a correlation, you will not understand what's happening. The minimum what you need is you need another opinion, a second opinion. Luckily, for about almost a century now, there is some solution, which is called corollary discharge or efference copy. This term means a lot of things to many of you, but the fundamental things are pretty much the same. Namely, every single part of the brain that sends out an action, whether it is a motor command, a autonomic system command, a endocrine command, or in fact just a thought, which is also an action, will send information or neuronal discharges to the relevant sensors. So now the relevant sensors, in this case V1 neurons, will have two information or two types of information. One is what is the world doing and what I am doing. And that's a very useful grounding mechanism. This is a philosophical problem of grounding, because there is something that is better known, that's the grounding thing, can be associated, can be linked to something that is less known or unknown. Only this way I can know that I am the agent of my action, and only through actions, through movement, we will learn that a stick in water is not broken, because we can stare it visually, this picture forever, and we will not get smarter. But the moment I start moving the straw around, in an instant, I will know that it's one unit. And this idea about the corollary discharge goes all the way to complicated things such as perception, such as thinking, and even, I claim, perhaps language. And this is maybe where Julius is agreeing with me, or there are many points that we disagree. But this is the fundamental mechanism how we can go from simple circuitry to more complicated circuitry in evolution. So let's move to the next bigger topic, is that now how do cognitive mechanisms can emerge from this fundamental simple mechanism? Let's look at the simplest, most simple hypothetical brain, whose goal is to generate an output and learn the consequences of its output. The most important thing for me that the brain can do is to learn about the consequences of its own action. In other words, it can make predictions, but in the prediction, the agent, that is the body and the brain involved, is present. So this organism can do it pretty well in a very simple environment, and where the input and outputs are also condensed in time to each other. So what happens over the evolution is that this little circuit that we have here will get a little complicated. It gets complicated in a sense that you have a thalamocortical loop, then you have a hippocampal loop, you've got a cerebellar. There are many, many loops based, built upon each other, and then the input outputs will be more and more and more and more complex. You know, in AI terms, you want to say, okay, I put deep layers here, and these are the deep layers between the outputs and the inputs. And of course, this is already present, the corollary discharge, but it's even here, it's more sophisticated than in a cricket, or in a frog. Now, this brain has the same goal, which is to predict the world and to predict its own actions. But it can function in a very complex environment, as well as it can function also at a much longer timescale. This kind of a brain, such as yours and mine, can perhaps evaluate my plan to decide whether I will go to college or not. And that I can evaluate potentially what will happen 5 years from now when I finish college, or 4 years from now when I finish college. But this is not enough. But there is a critical thing that I claim is necessary for cognition, which is at some point, these loops are getting so smart that you no longer need the world. In fact, what happens is that what you see and what you feel is, of course, not what comes to your retina, but what you see is the computation in your own brain. When I close my eyes, the pictures that I see here on my screen, I see some of you at least, don't disappear. And the objects around me don't disappear, because what I'm seeing is not exactly the instantaneous photons on my retina, but an integrated picture, and that seeing itself is the brain computation. If you believe me that that's the case, then now we can indeed remove the world. And then we can do 50 fictive explorations. We can call it, or in psychology, you call it the what if scenarios. What if I do that? What if I do that? And evaluate the potential consequences of my action. So once you have this complicated brain, which is self-organized and is capable of functioning without the world, then this is where we can start thinking about things like memory, planning, imagination. And you will see these different words are not so different from each other, because the computations that underlie these three words are probably pretty much the same. So let me give you an example of this bigger framework. Here is an example. The example comes from the world of the hippocampus and entorhinal cortex, or the navigation devices of the brain, for which Nobel Prizes were given not so long ago. And it has been known since 1971 that indeed the hippocampus and its allies are serving spatial navigation, which comes in two flavors. The simple one is what we call dead reckoning or self-referenced navigation, which is a very simple thing. If I would be in person at the University of South Carolina and I would be in a lecture room, then Julius could switch off the light and so on, but I could still find myself around because I could walk, and I have idiosyncratic memory, and I could walk back to the same place where I came from. I can do the same walk again in a different direction. I can come back to the same object, and I would recognize that, this is the same object that I intersected now from different directions. And then I have a node. And if I do this exploration for further and further and further, then I will have several nodes, the layout of the room that I'm exploring blindfolded. And I will have a pretty good idea about the relationship and distances because I measured them through my actions. And then I can make a map. Now, when I make a map, then the map is on a piece of paper. I can give it to anybody else who can read that map or learn how to read a map. And therefore, what happens here is that I study something, and this is called egocentric action or first person action. And all that experience that I have is mine, but then I translate it into something that is independent of me, just a relationship among the objects in the world, and this is becoming a map. And both of these processes are performed more or less by the hippocampal entorhinal system. If you want to be very strict, you would say, these are the place cells, and this is egocentric navigation is done by the hippocampus, whereas the map base is done by the entorhinal cortex, but of course it's not right. The two of them are working as a unit. Now, for about the same time, since 1971, and especially starting out with the famous patient HM, the hippocampus has been advocated also as a memory device, especially for what we call a memory that we can verbally declare, declarative memories that or consciously declare. And of course, this right side was studied in different laboratories, mostly by human psychologists. And they also divide the declarative memories into two types. One is what you can call episodic memory. The other one is semantic memory. Now, episodic memory is a really egocentric one. We are the collection. Our personality is nothing else, just a collection of our personal autobiographic memories. And the way it works in all animals is that we have the experience first. For example, the first time you see a dog, it's an experience. It's an episode. But then you see another dog and another dog and another dog, and then you see a lamb or goat, then you call it a dog too, because you generalize unless there is a reason to make it separate. And then the conditions that led to the individual episodes are stripped off, and only the term dog or dogness remains, and there's semantic information. So note that this is the same kind of thing that happens on the left side, that one is personal and it has time and space and so on, but then the other one has only relationships. And that's the same case on the right. So we can make the generalization is that initially nature worked out a mechanism for the animal, or for animals to navigate around the world with the help of the outside cues. But the system became very smart over several millions of years, and then it no longer has to rely on outside cues because it can do a navigation mentally. This is what we call mental travel. We can go forward or backward in time. So this is the current thinking, at least, why the same structure can be claimed by one group of people, that's a navigation device, and by other group of people as memory, because the two of them are fundamentally the same. The hippocampus runs exactly the same algorithms when we are doing the two types of solutions or two types of tasks. The only difference is that in one case we can rely on the outside world. In the other case, we don't have to rely on the outside world. So this was very beautifully specified by Enver Tulving. Many of you may know his name and his extraordinary work that he has done. And he defined episodic memory for us the following way. Episodic memory is basically what happened to me, where and when. And this was an enormous blessing for most neuroscientists, because now we had a roadmap, what we have to do when we go and try to understand brain mechanisms of episodic memory, which is figure out where the what is coded, figure out where the where is coded, and figure out where the when is coded. And this is an enormous simplification compared to the requirement that we would have to deposit and store every single experience in our lifetime, because that would be an enormously long list and there would be no classification, and it would be so difficult to retrieve from this long string of items or events. On the other hand, if we have now 3 axes and we are breaking up the problem into 3 axes, things are simplified, because if we code the where and when and what separately, then all you have to do during recall is to get one of them, 2 of them, and then multiply the marginals, and the coincidence of what, where, and when will give you the here and now. So looking at this coordinate system, you can say, aha, we have a solution now and definition of what is an episode. Episode is a trajectory, such as this yellow one, of an event that happens in space and time. So this is the series of what's, what happened on my birthday party, who arrived first, who came afterwards, what happened when the glass was broken, and then the dog came in, and so on. So this is an episode. But then a similar episode can happen, and another episode which has a common node, just the same way as explained for navigation. And this node is becoming the birthday party, irrespective whether it was mine or somebody else's. That node, of course, is frozen in time and frozen in space. In other words, semantic information can happen in this, according to this model, when you strip off the context, the spatial and temporal context, and you just have the abstract out the thing that is common to all of it. Now, I have to put something in parentheses here, and they said this is true for every animal. In humans, we've got a shortcut for semantic information, because we can learn semantic information without our own experiences from our caregivers and from our teachers and from books and so on, because you can just tell me that this is a mouse, and I look at it and I accept your words and said, okay, from now on we will call it a mouse, because this is how I can move my cursor on the screen, without ever asking why this is called a mouse and how it came out to be called that way. But in all other animals, the way I would say the simplest and most common way of getting a semantic knowledge is through our own episodic memories that intersect with each other. So this is cool, and we said we have a roadmap, but there is a little problem here. The problem here is that this whole idea is based on Newton and Immanuel Kant. It was Newton who defined space as a theater, it's a grand container into which we put in our things. And this is exactly what Enver Tulving did. They said, okay, what happened? These are the ideas, the items, or the events. We can put them in the container, which is space, and we can put a stamp, which is a timestamp to it. And this is how it works. But if it turns out that the Newtonian mechanics does not apply to the universe, and it may not apply to the brain either, then we have a problem. And the problem is summarized here by this extraordinary physicist, Carlo Rovelli, that there is no longer space in physics or for physicists that contains the world. And there is no time in which events occur. So where do we go from here? So I will guide you through a couple of slides, and I will give my answer to you. So these are the elements of navigation. And I assume most of you know about it, because you could read about these in New York Times in 2014. So we have head direction cells in the brain. There are grid cells in the brain that give you a map-like grid that we can relate things to each other, and then we can put ourselves on the map with the help of the place cells, and then we can calibrate the map and so on. And this is the metaphor that we are working with. Now, of course, the interesting thing that when you use these metaphors and you ask the important question is that why neurons in a rodent's head or in your head generate a sequence that is, they are firing here, there, and there is a series of activity that one set of neurons go to another set of neurons happen. Then one way to say that, the reason why it happens this way is because the world, the outside cues, drive the neurons in the hippocampus. Again, this is a Newtonian idea that you have to move something, and the mover is always energy that has to come from somewhere. It cannot happen from inside. So the reason why P1, place cell 1, place cell 2, place cell 3 are happening one after the other is because when the animal moves around, then there is a corner, there is an object, there is food, there is many other things. But if this were the story, I couldn't figure out how can I give this presentation to you today from my head. Because the truth is that I have slides, but even without the slides, that is without any help from the external world, I can give this talk. And the reason why I can give this talk is because when Julius introduced me, I had an initial condition. I know that where I am in at University of South Carolina, and my job is this. And from then on, an assembly sequence happens in my brain. The first set of cells will give the information to the next one, and next one, and next one, and next one, and it goes on all the way through my lecture. So there is another way to think about it, namely that there is the reason why these sequences occur is because there is an internal dynamics. We don't need a Newtonian driver. We don't need the external world to drive our brains. The brain is a self-organized system, and this internal dynamic happens whether you want it or not. So how do we test this? Well, we tested it in a relatively simple way. We chose a hippocampus dependent task, which is a T-maze, where the task is to go to the right. And if you find water here, then remember that I came from the right, now go to the left. It's called the spontaneous alteration task. The trick that we used is that between the choices, we had a delay. And in the delay, we asked the animal, this rat, to run always in the same direction and with the same speed. So what we have done in our power, we have done everything that the external world remains constant from the outside and from the body. Now, because the animal's head doesn't move, the prediction from the navigation theory, from the map-based theory, is that there will be always a set of neurons in the hippocampus which will fire forever as long as the animal is staying at the same Cartesian XY coordinate. On the other hand, if you believe me that the hippocampus just cannot help, it always generates sequences, then this rule will not be the case. And in fact, this is what we found. We found that instead of finding neurons that would be firing forever, which is continuously from the beginning to the end of the run at the same firing rate, and everybody else would be completely silent, only the subset of cells that determine the XY coordinate should be active. Instead of that, what we found that all neurons were active sometimes along the journey. So what you see here on the x-axis is time, how long the time an animal has to run between the left and right choices. Every single line is a neuron here, coded by its maximum firing rate, and then we ordered it according to the maximum firing rate, and you can see that this is many neurons here, they tessellate the entire journey, that everybody is active at a certain time of the running. Now, the good news is that this trajectory, which is repeating and repeating in every single trial, because the initial condition is the same, and the initial condition determines this complicated trajectory in the n-dimensional space of the hippocampus, is unique to this initial condition. But if the other condition applies, that is, the animal is making a future right choice, then you can see from the top and the bottom, you can compare them, they are very different. In other words, one trajectory going to the right and the other trajectory going to the left are so different. And they are so different and that we can make a prediction 15 seconds earlier while the animal is running, which direction the animal will make at the end of the central arm with 90% accuracy, including all the errors. So this is extremely interesting and powerful that we can make these predictions pretty long. Now, are these place cells? Well, they are place cells in an interesting way, of course, because they are determining every neuron in the hippocampus in this particular paradigm, how we set it up, determines the same exact XY coordinate. So they cannot be. But then what else can we learn about this? Well, what you might have noticed that instead of talking about distances that is happening when animals in the maze, in this case, the X coordinate was in seconds. And in fact, what we can do is looking at this sequence and ask the question is that if I see these neurons firing one after the other in this time window or in this time window, can I tell with some reliability how long, how many seconds the animal have been running from the beginning of the journey? And the answer is pretty good. I can make a pretty good prediction, even if you have only about 100 neurons. If we had 500 neurons, 1,000 neurons, then the prediction error would decrease quite a lot. So out of the 15 seconds, the error is about 2 seconds only. And this animal had the fewest neurons. And if we had more neurons, then probably the error would be down here. So that's interesting now, because it's not only the distance that may reside, or the space may reside in the hippocampus, but now also the time. And remember, we are looking for the mechanisms of the what happened to me, where and where, and it seems that the hippocampus is a good place for it, because now we have the what, which is which direction. We have the distance, because everybody told us that the hippocampus is a spatial device, and now we have the time. So much so that a good friend of mine, the late Howard Hickenbaum, took this so seriously that these cells, when animals running in a wheel or running in a treadmill, and the sequences occurred, he called them time cells. And it turns out that every single cell sometimes could be a place cell, but the same exact neurons sometimes can be a time neuron. This is not exactly what he said, but this is my summary. So that now we have an interesting problem. The interesting problem is this. The only measurement that I or the experimenter makes is this, a sequence. From the sequence, I can predict the what, because I know whether the animal is going right or left. I can predict or tell you how many centimeters the animal was running, that is the duration. Or I can tell you how many seconds, that is how long the animal has been running. So what happens here, we have one measure and 3 names. We call them by different names, but it's exactly the same. Now, let's examine this a little bit deeper and say what happens in the mind of the animal, if you believe me that mice and rats have also their minds, and especially what happens when they are conscious or subconscious. So here is a little movie. What you will see here is many neurons recorded simultaneously. I stop here for a second. What you see is a sequence here. The blue neurons fire first and the red neurons fire fast. On the bottom, you see a very fast oscillatory patterns that I called hippocampal sharp wave ripples. This is the most synchronous pattern in the mammalian brain. As you can see, many neurons fire simultaneously. So this is a very strong signaling mechanism by which the hippocampus can instruct the rest of the brain, neocortex and other structures, that something happened, and listen, now there is something coming out from the hippocampus. So I put this sequence here, just as a reminder for you. And then the animal is still sitting here, is not doing anything. Now the task is to go to the end, and the other end of the track, the animal will be rewarded. And listen what happens. So notice the animal is running now. So the animal stopped, rewarded, is drinking water. The theta oscillation is disappearing. There is no theta now. There is a little firing. And now we have seen another ripples, sharp wave ripple, and another very powerful burst that corresponds to this ripple. But notice what happened, that at the beginning, before the animal started, the plan, if you want, was played out to the rest of the brain. From the hippocampus said, this is the sequence of the neurons that will come up one after the other while I'm making this trajectory, while I'm walking through this space. So the sequence of the neurons, these are the place cells, are the same that happened during the sharp wave, and this is the same direction. But at the end of the journey, same sharp wave kind of same happened, but now the sequence is going backwards. So in the next slide, I show you the condensed version of this figure, namely that this is the sequence that happens on the maze itself. This is the forward and this is the reverse one. Now, we can call this one on the left planning or prediction of the future. And on the right side, we can call it postdiction, or you can call it memory. The animal recapitulates those places that it visited. And in fact, one of the theories I put forward is called the two-stage model of memory, where we made the claim that this is the important pattern for consolidating memory that happens typically during sleep. The interesting thing, of course, is that it's the same mechanism that is responsible for this one and this one, which we call planning and memory. So it is probably not very different from each other. And in fact, over the past decade or so, many laboratories, starting with Dan Schechter and Maguire in University College London, showed that those structures that have been traditionally used in fMRI experiments and were claimed to be the sites or the structures related to memory processing are exactly the same ones that are lighting up in human imaging experiments when the patient is imagining or planning forward into the future. So these are different words, but they may be supported by the same exact mechanisms. So where does this lead us? It leads us to a little thinking, namely that it doesn't matter how we call these cells. The brain doesn't care about names. What matters for the sequences that we just seen is how the downstream reader mechanisms will classify. Whether it is called one way or another, it doesn't matter. And this is an important thing, is because it seems that whether you call something a time cell, a space cell, a sound frequency cell or memory cell, it depends mainly how you set up the experiment and what variables you exclude and what variables you allow to happen. So that it seems that the hippocampus is a sentinel which is watching the activity of the neocortex. Whenever something comes from there, it checks whether it is novel or not and does the computation and sends back the answer independent of the modality. The hippocampus is a modality blind structure. So if that is the case, we don't call it a navigation device or time cells or place cells, but something else. But we have a problem now. Remember that we said, well, the hippocampus is not computing time. The hippocampus is not computing space. All it does is generate sequences, and the rate of sequences is determined by some other factors such as running speed or attention. So if we take out space and time from the definition of episodic memory, then what is left? So what is left is that we can say the hippocampus is a general purpose sequence generator that is not really containing the information about the world, maybe just a little bit, but the real knowledge is all in the neocortex. But the hippocampus helps to concatenate this in a logical sequence. So with some metaphor, we can say that the hippocampus is a librarian, or you can say it is a search engine that is not knowledgeable about the details, but the librarian can tell you where to find that information in the library itself, which you can imagine is the neocortex where all the knowledge resides. So if you view the hippocampus this way, then you can say, aha, the hippocampus is generating the sequences, and that's its main job. And this is, of course, the fantastic thing about episodic memory, because it happens in a sequence, it happens in a trajectory that you call spatial temporal context otherwise, but now we can toss it out and just rely on the brain mechanisms, and then points to the relevant areas and relevant modalities in the neocortex. And this is how we can compile, or we can put together memories, as well as how we can retrieve memories. This is a cartoon. If you're interested in further details, then I refer you to the original papers. Now, if this thinking is good in a sense, and we say that the sequences that are generated in the brain are not imposed from the outside world, but it's generated inside, then we would like to know what are the mechanisms of this sequence generation, and does it matter whether the trajectories go in any way, or they are already confined very powerfully by certain mechanisms, such as skewed distributions. I have written extensively about skewed distributions if you are interested in, because everything in the brain is log normally distributed that I know of, almost everything, rather than following a normal distribution. And this right cartoon here, it shows that this organization already puts a lot of constraints how the brain can function. And the constraint we can say is that it is not necessarily the way how we think, that the way how we learn, we put in a blank slate, and then we change the connections and so on. In other words, the brain is not a equally distributed system that is governed by simple Hebbian plasticity, but in fact is the opposite. The brain is a very rigid system. It can generate lots and lots of patterns, even if you have zero experience. So learning in this world is not a build up of a circuitry, but choosing a pre-existing circuitry. Now, you may say, oh, this is trivial or maybe useful, but nobody believes in the tabula rasa, but it's not quite right. Here is a quote from somebody whom you all know, Alan Turing, who is, of course, a giant in the AI field. It says, the child brain is something like a notebook, that is a tabula rasa, as one buys from the stationers. And of course, the entire field of AI follows his recipe. Nearly all current AIs are built upon the tabula rasa framework. And you build it, you train it, and the training, everything comes from outside. Outside, the self-organization is not something that is thought of yet. In the brain, self-organization is the most important thing that exists. Again, we have to not only speculate, but show this that happens with experiments. So here's an experiment quite some time ago that we have done with two of my postdocs, a student and a postdoc, and it goes like this. When we record from the hippocampus region, in this case, 2 place cells, and this neuron will fire at place L and the other one at the place B. The argument is why they do that is because they receive a lot of inputs from other place representations, in this case, the upstream neurons from CA3. And because these neurons, the blue neurons, corresponds to place A, then this neuron will fire. Neuron B will mostly connect it to ensembles that represent place B, and this is where neurons will fire. And then we also know that they receive inputs from many other places, but the reason why at place C the neuron doesn't fire is because the inputs are too weak, but they are there. So what if we change the synaptic weights between these neurons by artificial means, such as long-term potentiation? This is a phenomenon that we can exploit and do, and this is exactly what we did. As a result of what we did is we made new place fields, such as shown here, and we made place fields disappear. So in this case, there is a new place field here, but if you zoom in, or if I zoom in, you can see there is a little ghost here. So that cell already existed a little bit in a weak form. So we didn't make it, we just uncovered it or unmasked it. And to show that this is in the case, what we have done is that we looked at the relationship between the firing rate of the place field within this place field, what is the best firing rate and how it is tuned. Usually a tuning curve is better for a neuron, if the neuron fires a lot of spikes within this field. And what we found is that if the neuron had a very good place field, it didn't change by our drastic perturbation, but many neurons that were weakly expressing their place fields could be affected very strongly. In other words, these low firing neurons are very plastic. The fast firing neurons are typically rigid. So it means that we couldn't impose our external perturbation equally to the entire system because of the rules that already pre-existed in the hippocampal circuitry. Now, these two guys went on and they have established their own labs, and the same rules were discovered over and over. So this is an example from Ken Harris's lab, where he has explored the entire auditory cortex and showed that during sleep, the possibilities that exist in the interactions are very, very large. And in fact, when you perturb the auditory neurons with natural sounds, with tones or with anything, all of those patterns will fall or occupy the space within this larger realm of space that are played out spontaneously during sleep. George Dragoi went to work with Susumu Tonigawa, and he did something different than in the hippocampus, but fundamentally is the same idea, is that he put an animal into a novel environment. This is the animal is living in a home cage, sleeping, and then next day, the animal is put into the novel environment, and in a novel environment, there will be a sequence of neurons, just the same as I showed you. And then it puts the animal back to either rest or puts the animal back into the home cage again, and there will be a replay of the sequence again, the same way I showed you during hippocampal sharp wave patterns. The interesting thing is that when you take a template after sleep and you go back to the pre-sleep, they are very similar. In other words, I can record from a bunch of neurons in your brain today, and then you will listen to a talk tomorrow, and I can make a good guess which neurons will be firing while you are listening to the talk from recording from the night before. It gets a little spookier here, but given the time, I think I have to skip this. So I just show you a current incarnation of this old experiment with an updated method, which is optogenetics. What we have done here is that we chose a site in the hippocampus that we can stimulate with optical methods, that is neurons that have the genes for light sensitivity, and we can make artificial place fields. So under this electrode, with this neuron size micro LEDs, we activate a set of neurons, in this case, this neuron, and you can see that these neurons under the electrode shank will be active. But the cute thing is that they will be active not only during the time when you stimulate, which is this 3 trials, but persist after we don't stimulate anymore. I can go and skip these and this one also. Now we can concatenate all the new place fields that we generated. And he said, well, this is the center of the new place field we generated everywhere in the hippocampus. Let's see what happened before. So this is the first trial, second trial, and so on. It's 10 trial. Now for 3 trials, we do our perturbation, and this is the consequence of the perturbation. And now looking backwards in history, and we will see that these neurons were firing a little bit also before, which means that only those neurons which already had a pre-existing potential pattern can become or can be turned into a place field rather than any neuron at the will of the experimenter. Moreover, and this is the interesting part, we go back a day earlier in the home cage and we ask how these neurons which we manipulated relate to all the other neurons that we record simultaneously. That is the assembly partners of this neuron. Can the assembly partners of the neuron predict where the where the neuron will become a place field? And the answer is yes. Those neurons or those partners which we didn't stimulate, but were having a good place field at the place field of the future optogenetically generated neuron, can predict. Let me try it again. I confuse you a little bit. So from an assembly of neurons, we can identify where they fire on the track. And then we are recording at the home cage, and some neurons don't have place fields, but fire together with a bunch of other cells. We put the animal onto the track, and then we stimulate those neurons which didn't have place fields, and they become place fields. And they will become place fields where their partners already had a place field. In other words, the partnership, a pre-existing, a preconfigured partnership already predicts where certain things in response to the outside world will become active. With these and other experiments, I think we can simply give the answer that there is no such a thing as blank slate. It is a much more complicated picture that we have in mind, but at the same time, it's simpler. And this is a cartoon that you can say that the brain is basically, from the dynamic point of view, is a spaghetti. The hippocampus, but also the rest of the brain, can generate a lot of sequences, and they are available to be matched with experience. So when we learn, we are not making neural connections one after the other in a rigid way from de novo, because if we would be doing that, then brain complexity would be scaled with the amount of experience with what we have. And in fact, there is no evidence whatsoever that brain complexity changes no matter how much we learn. So in this world, it's not that neurons that fire together will be wired together, but it's the other way around, that neurons that have been pre-configured together will fire together, and learning is a matching process, a selection process, rather than a de novo build-up of connectivity. So the summary of this last part is I can imagine that a brain comes with a pre-configured dictionary filled with nonsense patterns, but those nonsense patterns, like a Chinese dictionary that I don't understand, but I can ground it to my known English words. And this is what I call a matching process. And learning is a matching process, or an unmasking between pre-configured patterns and experience. And of course, this is done by experience or something that is meaningful to the animal. And only this way you can make the selection. Now, everything is complicated and I used up my time. But if you would like to learn more about this, then Julius said that he already read this book twice. I suggest to read at least the introduction. Thank you very much.
Thank you so much, Yuri, for a great talk. And yes, the book is highly recommended to all who participate in this talk today. And of course, tell your friends. So thank you again so much for giving your lecture. We do have questions in the chat box. We have a couple of really lengthy ones. So I want to start with a short one that maybe that you can address. The first question is from Shlomit Ritz-Finkelstein. Would you please define action? Short question, possibly a really long answer.
Can I see you?
You should be able to see me.
So action is a short term for anything that is an output from the brain. And that could be control of muscles, that we usually call action. But for me, as I mentioned, hormonal control. And it turns out that the hippocampus actually has a very strong control on the hypothalamus, and through the hypothalamus, even our glucose levels in the body are being affected by hippocampal shockwave ripples. That's an action. And I call thought also an action. It's a deferred action, because thoughts are almost as useful as their translation into a future action. So anything that is this is the hand waving explanation. The other one is simpler. I can call I can use a neuroscience definition, is that any system that sends a corollary discharge to somewhere else is an action. So if we would be able to record from the brain the way how we can't yet, and we know that now the sensory inputs are getting input from other areas, I would call it an action system. And this is an interesting thing. If you follow today's literature, last year there were 3 papers in Science and Nature where there was a revelation that neurons in V1, primary visual cortex, are super sensitive to movement, and even a optokinetic reflex, which is you just move your saccades. Half of the brain is responding, whereas when you head is immobilized and you are immobile and you show visual patterns, only a very few neurons in V1 respond. So the brain cares a lot more about action than anything else.
Thank you very much. We have a question from Greg Hickok. He's been basically typing questions since the beginning of your talk, so he's hard at work. Action is helpless without perception. For example, large fiber sensory neuropathy would be a problem. Indeed, an action event is grounded in its perceptual target in this sense. At the same time, from an evolutionary perspective, perception is not much good in an organism that can't use that information adaptively. Why then should action be assumed to be primary somehow? Isn't it just the whole system that is primary? And the real grounding is not motor, but more generally a brain that can help replicate its organism's genes. There's a specific question that follows. If perception is grounded in action, how can a person who was born without the ability to control their vocal tract learn to perceive speech normally? For example, see Bishop and colleagues 1990. And also, if everything is grounded in action or movement, doesn't that predict that some people who cannot move, for example, in cerebral palsy, should not be able to perceive or understand the world?
Thank you, Greg. That's a long series of questions. I think you gave already the answer. Namely, depends on how you look. If you look at from the mind point of view, then things are complicated. But if you look at from the evolutionary point of view, then you will see that there are animals that have no sensors whatsoever, but they move. And if you are in an environment, in your own niche, where all you have to do is move around and the probability of finding food or bumping into food is high, then you don't need any sensors. I can put the other way around also, is as you said, that what is the point of perceiving something if you cannot act upon? And we have done a experiment published in Nature a couple of years ago, actually a long time ago, where we asked, what happens with a newborn? what is the first organized pattern in the brain? And it turned out that the first organized pattern is a spindle that is responding to a peripheral action, which is nothing else but release of acetylcholine to the muscles. So it starts with a stochastic stupid teacher, but the muscles react. And the consequence of this is that that area of the body that is activated will have an activity in the somatosensory cortex. Now, another area will receive the same stochastic pattern and it will respond, then another area will be here. Now, the interesting thing is that these muscles are anchored to the skeletal. So their degree of freedom is very much controlled. So those muscles that will in the future work together will have the same representation as a result of this action-based teaching. So this is one example. The second example is the other way around. You said, oh, how come what happens if you're paralyzed? Well, I had a cousin, and there is quite a bit of literature about this, that if you are born with a not cesarean section, but some other complications, in the old days, bones were fractured. And then what they did is they put cast on the limbs. And there are many old papers showing that that limb doesn't feel and doesn't respond. If it responds, it has a positive Babinski reflex, which is like decortication. So, and many parents are scared when it happens because they said, oh, we learned about decortication, what happens to this? And the surgeon will say, don't worry, we will move the cast and everything will come back. And this is typically what happens, that once the limb starts moving, then sensation comes back. So in this particular scenario, indeed, there is a good demonstration that without action, there will be no sensation. Language is I leave it for you, but that's a complicated thing, because language is not only about sounding, as you know, you can have various outputs, and that output communication can help you a lot.
Thank you. Yes, we wouldn't want to make things complicated here. We have another question from Daniel Chapman, which is related to the question just asked. One of the major aspects of your inside-out viewpoint is the persistence of internal dynamics in the brain. If you were to sever all sensory inputs to the brain, how long would these dynamics persist for? Is it theoretically possible that internal dynamics of an isolated brain would persist forever in the absence of sensory input? Or do you view sensory input as a novel dynamic that tips the scales of imbalance in brain dynamics, and that it, the brain, would eventually reach an equilibrium without input? For argument's sake, let's assume this is a fully developed brain of an adult human or other animal.
Well, the short answer is, hi Dan. My short answer is that it's forever. And the simple answer is to say, well, you fall asleep tonight. What is Vivan doing? And we just published a paper 2 years ago, and we were shocked to learn that no visual person ever recorded neural activity in V1 during sleep. And then we found that the activity in V1, which is a primary sensory area, is the same level of activity than in the waking visually processing brain. In fact, tissue culture, and now what is very popular is the organoids, human organoids, brain organoids. They have their spontaneous activity forever without any external input. The brain just cannot help. It generates constant activity. And I make a Gedank experiment. If you would be removing all your sensors, all your sensors will be connected to your brain, but you would remove the action ability, that brain will never learn about the world. You don't learn about parallax, you don't learn about stereopsis, and so on and so on. But you're asking the other way around, is that what will happen if you sensory deprive yourself? It will be a funny feeling. For $250, you can do this experiment by floating in the salt water, and then you plug your ears, and it's complete darkness and so on. But nobody measured the brain activity. I bet it will be exactly the same. Yogis do it all the time. They try to reduce their inputs, and their brain activity just going on forever.
Thank you.
You can try it in blind people. They have huge alpha activity, huge alpha, even though even if they are blind from birth.
We have another question from Shlomidrit Finkelstein. We surely must translate between space and time, since we have no sensory mechanism for time. Indeed, often in cultures, people use such exchange. For example, the song lines of the Australian Aborigine for spatial navigation, or the 3 cigarettes answer that often a Bedouin would give when you ask how far a place is, meaning it is as far as it takes to smoke 3 cigarettes. It's a comment, I guess. Yes.
Yeah, it's thank you for the comments. Indeed, half of brain languages don't have tense. Chinese don't have tense. Hungarian operates very differently than the languages, and space and time are interchanged. I say, oh, the Starbucks is 5 minutes from here. I don't say the distance, but we translate back and forth and so on. And by the way, this is the same exact thing, that when you are driving your car, then you ask how far is your work, and then my answer is that it's 1.2 million strokes, because that's the only thing that matters in the car. But that could be translated into distance, that could be translated to time, but it's one mechanism. So I fully agree with you that this is a framework. We cannot talk as humans. The reference axis of our thinking is space and time, but that doesn't mean that we have to search for mechanisms inside the brain to find those representations, because they are all human. And how do I find, without measurement, without an instrument, time is not a scientific concept. But in a rodent brain, rodents cannot read the clock. Rodent cannot read the rods, distances, and so on. So thank you for the comment.
In the context of a lecture in the C-Star series, we really don't want to start measuring time in terms of number of strokes, György, but because that would be a morbid thing to do.
Exactly, because we are simplifying it to have an agreement. This is why we need units. I said 1 hour is your time.
Dorian Pustina asks, the evidence and the analogy of the hippocampus as a librarian reminds me of a center of consciousness. What is the relationship between hippocampus and consciousness in your view?
We have to spend some time with you and define consciousness that we can agree upon. But having said that, so HM and people who have bilateral hippocampal removals or damage, are they conscious? And it depends how you define it. But if you say that is the agent is present, and when you ask them to narrate and say what happened to your 15th birthday, or what happened when you finished college, or something like that, HM could never narrate. He could give you fragments of information, but couldn't put together the sequences of their occurrence. He was absent, what Enver Tulving calls the autonoetic. That is, I am part of that story. That's completely missing. And if it's that completely missing, and if you make the claim, not me, psychologists made the claim, that we are our memories, our personal memories, and we cannot recall our personal memories, are we conscious in the wide definition of the word? I can interact with you, but this is where the problem with consciousness starts. Is the minimal consciousness? Is the how do we define it? So in this broad sense, yes, the hippocampus is super important.
Thank you so much, Yuri. We are going to end it here. We do have a few more like comments back and forth, going back to the responses, but I'm sure we could spend another few days talking about your interesting topic. We thank you again for your talk, and thanks to the audience for all their questions. We hope to see you again in 2 weeks' time for Yasmin Faruqi Shah, and then of course next season. Thanks and bye-bye.
Thank you, everybody. Take care.